Hello everyone. In our ongoing series in biotechnology, today we are talking about the production of cloned and transgenic animals. The methodology involves the vector-mediated gene transfer, and we discuss about the retroviral vectors to start with. Now, retroviruses, as we all know, they have RNA genomes that are used as templates for reverse transcriptase to synthesize a DNA copy that can be inserted into the host cell genome. Now, not just retroviruses, but any virus-mediated gene delivery system uses the cell receptor recognition system of the virus for binding to a specific cell type, which is followed by the internalization of the vector DNA and its transport to the cell nucleus. So, the same is the case in bacteriophages, where the bacterial viruses or the bacteriophages, they are inserted into the bacteria. In the same way, the animal cells also take up these viruses and they involve the cell receptor recognition system of the virus, where the binding occurs, the internalization occurs, and then the transport into the cell nucleus. Now, once transported, this recombinant DNA, in our case, or the vector DNA, can be integrated into the host genome, or they can still remain as the extra chromosomal entity. It depends upon various viruses. Now, since high efficiency of vector DNA transfer is important, the administered sample of viruses should contain mostly vector viruses with very few and preferably no infectious virus particles. Now, this is again very important, because when we use these viruses for gene delivery system, we want to make sure that the administered sample of viruses should contain mostly the vector viruses. So, the viruses, they have been rendered their infectious nature has been rendered inactive, and these viruses we intend, because we want to produce more amount of DNA or high efficiency of the vector DNA transfer, we ensure that only the vector viruses are transferred and there is no infectious virus particles that are transferred there. So, for that purpose, we utilize what are called as the packaging cell lines. So, I am talking about the retroviral vectors and how they move, and then we will see that how they are then used to create the transgenic mice. So, what are these packaging cell lines? These packaging cell lines are cultured cells carrying genes that express viral proteins that are necessary for the formation of virus particles, but are not capable of producing replication competent viruses. So, now it is very important, one virus which we are using to introduce our vector, the recombinant DNA, into the host cell, we would like that virus, for the high efficiency of the transfer, should contain only the vector DNA or majorly the vector DNA. But if we want to make more copies of this vector DNA, we would want to harness the life cycle of the viruses. So, the virus particles need to be also formed. So, for that, what we use is that we infect the host cells along with what are called as the packaging cell lines. So, we also use these packaging cell lines, or either use the helper vectors, what they are called as, or we use packaging cell lines. These packaging cell lines basically carry genes that express the viral proteins. So, the viral vector carrying, they only contain the viral vector, they do not contain for the formation of the virus particles, but the packaging cell lines already contain these viral proteins. But the important aspect is that they are also not replication competent. So, they are not able to produce the infectious viruses, but they can, if the complete set occurs when you introduce the viral vector from outside, then they can produce the entire virus particles, which are again not replication pro. They are replication, they are not able to produce the replication competent viruses. So, this is again very important, and we will see when we use the viruses, it is a very important for biological containment as well, that we need to have replication incompetent viruses to produce, because otherwise they will replicate and infect any other cells, which will be again a very which would be a biological hazard. So we need to contain them also. So that is why we use these cell lines which are not capable of producing the replication competent cells. However, they would give the proteins which are necessary for the formation of the virus particles. After transfection of a packaging cell line with a vector construct that is equivalent to the length of the wild type virus genome and that carries the appropriate DNA sequence for packaging into a virus particle, the input DNA is replicated, assembled into viruses and released into the cell medium. What does this mean? That what happens is that we transfect these packaging cell lines. So what are these packaging cell lines? These packaging cell lines contain the genes for the viral packaging. So the proteins for the formation of the virus capsid, that is what they contain. But when you transfect them with a vector construct, now what is this vector construct? This vector construct is carrying the virus genome as well as our gene of interest. So this has been created in the vitro. So in vitro, this vector construct is created just by using the routine recombinant DNA technology we have talked about earlier. So the same way this vector construct is created and then transfected into the packaging cell line. Now you will have a complete set where you have got the RNA, the genome which is produced from this vector construct and the packaging proteins which is produced by the packaging cell line. So you have that which is equivalent. Now important aspect is that we have discussed in detail about the selection of the bacteriophages etc. And we talked about that the packaging can only be done if the viral genome is of a particular length. That was one of the selectable marker, that if it is of appropriate size, the viruses particles would be packaged. So equivalent to the length of the wild type virus genome and that carries the appropriate DNA sequence for packaging into a virus particle, the input DNA is replicated, assembled into viruses and released into the cell medium. So that is how the new viral particles containing the recombinant DNA will be released. The non-infectious vector viruses are concentrated and then prepared for the use. So these vector particles, however, would be non-infectious vector viruses because they would not contain the infectious part which is required. So that, and then you can use these viruses. These will be now the recombinant retroviruses. You can use these viruses and introduce into the animal cells to create the transgenic lines. So this is how it understands. Let's look at the diagram to understand it more. Now this is a retroviral DNA, for example. This is the retroviral DNA. This is carrying this gene, which is gene X, is our transgene. And it will contain some selectable marker. So this is done in vitro. This is the construct that is made. So this has been made by a recombinant process. If it is a retrovirus, you have done it in the RNA genome as well. And then you can convert it into a DNA molecule by using the reverse transcriptase in vitro. Or you can introduce the RNA, but introduction of DNA is much easier. Or you can introduce the RNA and the reverse transcriptase can then convert it into the DNA molecule. So whatever way, this is the genome, the retroviral genome, which is carrying the gene of interest, selectable marker, and then there are two repeats. There are long terminal repeats at the end. Now these long terminal repeats are the repeats which are basically causing the transcription of the mRNA, which will be there, and then it will be converted by reverse transcriptase into the DNA. So in the vector you can create such vectors and you will take a cell line. So now you can see in the cell line you have got a part of the retroviral genome, and this part of the retroviral genome contain 3 genes on separate segments. One is the GAG gene and another are the POL and the ENV gene. So these GAG genes are the group of antigen genes. They are polyprotein, they would produce polyprotein molecules. So these polyproteins are present and they are very, very important for the generation of the core particle. And then they will also contain polymerase. So this polymerase is the responsible for the synthesis of the DNA molecule, which is the reverse transcriptase, and you will have the envelope protein. So these are again required for the generation of the envelope. So GAG and envelope, GAG and ENV proteins. So GAG, Pol and ENV proteins. The packaging cell line contain these packaging proteins. However, it lacks what is called as this omega. This is an encapsulation protein. This encapsulation protein is provided in the vector, you can see here, but not in the packaging cell line. So it can produce these proteins, but it cannot package the viral particles. So they can produce. So these are therefore termed as the packaging cell lines. They have been manipulated so that they only contain the packaging proteins but do not provide the encapsulation protein, so the viral particles are not formed. Now when you transfect these retroviral DNA, which is containing our recombinant DNA molecule, our gene of interest, you transfect into this packaging cell line. So when the transfection occur, you can see that from here it will go inside, and now this forms a complete set. Not the non-replicative set, but it forms the set where you have got GAG, Pol, ENV genes as well as a omega gene which is causing the encapsulation. These long terminal repeats are producing the RNA and the polymerase is converting it into a DNA molecule. Now that DNA molecule will now be incorporated into the viral particles. So the viral particles will be formed just like this. But they will package the gene which is like that. But now again they do not contain these genes. Now because these viral particles do not contain these genes, when they are going to, they are not able to replicate by themselves. They are not able to replicate, and therefore they are non-infectious viral particles. We can concentrate these viral particles and introduce it into a host genome where this particular gene of interest can be integrated, and we can then check its expression, but by itself these viral particles cannot infect any host cells because they do not contain the complete set that is required for the infection. They do not contain the infectious a part of the genome from here. So I hope you understand how these retroviruses work. So you take the packaging cell lines, you take the retroviral construct, the viral construct, the vector construct containing our gene of interest, which can be transgene, and then you introduce it and you create these particles. You can then concentrate these particles and use it. So these particles can then be concentrated and used. And how will they be used to create transgenic mice? You will have a donor mice. This donor mice is going to give this embryo, the early stage embryo, 8-cell stage embryo, 4-cell stage embryo, and then you will introduce the transgene containing retroviral particles. So these retroviral particles have been generated in this manner. These retroviral particles will enter these growing embryo cells by the cell receptor specificity that we talked about earlier, which is the case in any of the viruses that is required. So now these retroviral particles containing our gene of insert will enter or will be introduced into this embryo by harnessing their receptor cell type specificity. They follow the normal infection route that they are going to be introduced into. So they can be introduced into these cells, or they can be used various other methods, generally because they are non-infectious particles. So basically what we do is there is certain kind of a cell type specificity here, and then they can be entered, or you can also use certain physical methods to introduce these retroviral vectors inside these things. So what happens then is, then that you create this kind of this construct, you put it into a foster mother. So these foster mothers will then give pups, and in the pups you can analyze and check where the transgene is expressed. Now this transgenic organism will be called as the founder, transgenic founder. This will then be bred to produce a line that will contain these transgenic. The matings are carried out to determine which pups have the transgene in their germline cells. Transgenic lines can be established from these founder transgenic animals. because you are introducing at this embryo cell stage, then this vector could be introduced into any of the cells which could be prospective germ cells or prospective somatic cells. Now they are a ball of cells. During the development, they can form any of the different cells. But we want that these retroviruses enter into the dividing cells or the germline cells. So when they enter the germline cells, then we identify which of the pups will have these transgenic expression in the germline, and then we basically breed them and create a transgenic line. So these are the founder transgenic animals. So this is the mechanism or methodology that is involved in the creation of transgenic mice using retroviral vectors. However, there are certain limitations of using these retroviral vectors. The retroviral vectors can transfer only small pieces up to approximately 8 or 8 to 10 kb of DNA, that because of the size constraint may lack essential adjacent sequences for regulating the expression of the transgene. Now, when we want to introduce a transgene, then we want to basically express this transgene in these organisms. It can be for gene therapy. We'll talk about the application later, that gene needs to be expressed for in all the cells to create a disease resistant variety, or in all the cells or in some certain specific cells, for example milk producing the milk glands, so as to express the milk protein, or it has to be expressed, but the expression of the gene is required. Now the expression of the gene requires the promoters and mediators and enhancers, and there are various regulatory mechanisms which regulate the expression of a gene. So it depends upon where this has been integrated, but because there is a limitation in size, and if our gene is of that much of size, then we cannot insert these regulatory elements along with the genes. So retroviruses, there is a size constraint that is there, that limits the application of these retroviral vectors for the generation of transgenics. The genome of the retroviral strain or the helper virus that is needed to create large quantities of the vector DNA can be integrated into the same nucleus as the transgene. So this is again one thing which is there. When you are using the retroviral strain, the helper virus, so if you are infecting an organism, and when you are using the retroviral strain, then the helper virus genome, we want only the transgenic genome to be introduced into the animal cells, but in some cases the helper virus genome can also be introduced into the same nucleus. Now as you will see, if in the same nucleus the RNA construct, the vector construct is inserted, as well as the helper virus construct is injected or introduced, then it can produce the infectious particles, or it can produce. So the helper strain retroviruses could be produced by the transgenic organism, which is undesirable. We don't want the helper strain retroviruses to be produced. We only want the transgenic. We don't want these we want that only our transgene expression is there. They should not contain any contamination of the retrovirus genome, any contamination of that. So that can be a problematic aspect too. So retroviral vectors, they are good for producing the transgenic mice, one of the methods which is involved, but as I said, there is a limitation, the size constraint limitation, as well as the probability or the chance of creating transgenic animals producing the helper strain retroviruses, and which is completely undesirable. So some other vectors, the viruses that are used are the lentiviral vectors. These lentiviral vectors or the lentiviruses are again a group of retroviruses that includes human immunodeficiency virus, which is HIV virus that cause AIDS, and similar viruses from other animal cells. So human immunodeficiency viruses can also be used. Obviously their infective part is removed or they are generated in a manner that they do not produce infectious particles, but they act as the vector system to carry the transgene into an animal and into a growing embryo and produce a transgenic animal. So looking at the lentiviral vectors, the advantages of using lentiviral vectors are that they are not silenced in embryos. Now talking about the limitations in retroviruses, the general retroviral vectors that we discussed, one of the disadvantages is that they could be silenced in the embryos as well. So what do you mean by the silencing? Silencing is RNA interference. This we discussed in the sessions in molecular biology. So we have the complete set of molecular biology sessions on the CEC Gurukul site as well. There we discussed about the RNA interference, or the gene silencing, where the RNA molecules, the small RNA, microRNA molecules, they silence the expression of a particular genes. So in here as well, because the retroviruses, they can produce, they can be integrated at certain parts in the embryo where they are silenced. There are many different mechanisms for gene silencing as well, but they can be integrated where they become silenced. So lentiviruses have an advantages that they are not silenced in embryos. They are capable of delivering large segments of DNA into the host genome. They are stable for relatively long periods, so there is no loss of transgene during the subsequent breedings. They have low immunogenicity. So immunogenicity is again very important, because the retroviral DNA vector system, vector that you are inserting into the mammals, they act as immunogens. So you cannot repeat using the same thing in the same organism, because it will have an immunogenic response. But lentivirals, they have low immunogenicity. And the major advantage is that they can infect both dividing and non-dividing cells. So they can be introduced to express the genes in all the non-dividing cells as well, and not just the germline cells, but also the differentiated cells too. They can infect those cells. So lentivirals have many advantages which is there. Now looking at the lentiviral makeup, this is how the lentiviral looks like. So you have got a transgene. The important aspect is that it will carry a promoter. So it will carry a promoter of its own for the expression of this transgene. So expression is enhanced here. It does not require, it is carrying its own regulatory element, So it is not requiring the promoter of the genome to be expressed, the transgene, but it is carrying its own promoter. It contains these long terminal repeats at the two ends. These long terminal repeats at the 5' and 3' ends are required for the production of the lentiviral RNA, and the packaging signal, as we said, this encapsulation signal, the packaging signal is required for packaging the RNA into viral particles. You can see again it is not carrying any packaging proteins, but it is carrying the packaging signal. The packaging proteins will be as usual provided by the packaging cell line or by the helper viruses. But it does contain the packaging signal which is there. Then it also contains a polyurine sequence, tract sequence, and a Woodhock post-transcriptional regulatory element. These two elements are there. They enhance the transduction of host cells and increases transgene expression in the animal cells. So again, as I said, one of the advantages of lentiviral vectors, use of the lentiviral vectors for creation of transgenics is that there is a increased enhanced expression, and there is more efficiency in the transduction, and there is a stable transduction as well. So these are the two elements that helps in that. Then a regulatory element within the 3' LTR, this black dot, is deleted to prevent the production of vector RNA from a promoter contained within the LTR. Now this LTR is going to be expressed, these are going to produce mRNA. As I said, they are required for the production of lentiviral RNA. So this they produce, but they also contain their own promoter. So there is a possibility that this promoter is going to express the transgene as well. But we do not require that in this case because the transgene is carrying its own promoter. So we don't want that Therefore, this promoter system is basically deleted. This portion, the regulatory element, is deleted, so that this is not a regulatory element within the 3' is deleted to prevent the production of vector RNA from a promoter contained within the LTR. So we don't want that. Transgene is carrying its own promoter for the production of the lentiviral DNA. So lentiviral vectors have been used successfully to introduce a variety of transgenes into embryonic cells or early embryos of mice, pigs, cattle and birds, and these genes are highly expressed either ubiquitously, which means in all the cells, or in a tissue specific manner. For example, the production or the expression of milk producing gene, milk gene only in mammary gland. So that is a tissue specific expression depending on the promoter that you are using. So if you are using a generalized promoter, it will be expressed in all the cells, or if you are using a tissue specific manner, then those genes will be expressed only in certain tissues. So there are many advantages of using and applications of lentiviral vectors over the use of generalized retroviruses that we have talked today. So in the series, we are talking about the generation of transgenic animals, and generation of transgenic animals, the first method we have discussed today, which is by the use of retroviral vectors, and because these retroviral vectors have certain limitations, then we are also using, we also studied and understood how lentiviral vectors can be used to produce these transgenics. We continue with this method in our subsequent sessions. Thank you.